Okay, in this video, we're going to do an overview of what's called the cell cycle, which is basically the life cycle of a cell. So just like humans have a life cycle, we go from infancy to a toddler, to a child, to a teenager, to an adult, to old age. A cell has a similar kind of a life cycle, and there's names given to the different phases or stages of the life of a cell. And so this diagram we see here is kind of like showing, starting off with the cell. This is where the cell starts, right? Oops, excuse me. This is where the cell starts right here. And this is its life cycle. As it ages, it goes around through these phases, and at the end, it splits into two cells. So the life of the first cell is kind of done, and it's become two new cells. So these stages in the life of the cell have names, and you can see the names are some kind of phase. So, for example, up here we have interphase, and that's this phase of the cell here. And then we go from interphase to something called prophase, and then into metaphase, anaphase, telophase, and finally cytokinesis. So instead of saying toddler, baby, toddler, child, teenager, we have these names: interphase, prophase, metaphase, anaphase, telophase. So you may have already come across some of these terms, especially the prophase, metaphase, anaphase, telophase. It's known that those parts of the cell cycle are known as mitosis. You can see that word right here, mitosis. And mitosis actually specifically refers to just these phases here in pink. And when we think about mitosis, what we're really thinking about is a cell splitting or dividing into two. So we're going to go through kind of what's happening in these various phases of the life of the cell. And we're going to end up with kind of thinking about mitosis specifically, which are these pink phases at the bottom here. All right, so when I learned this material, I learned that the cell went through these different phases: interphase, followed by prophase, metaphase, anaphase, telophase. So we had this little acronym. We had IPMAT, IPMAT, which was interphase, prophase, metaphase, anaphase, telophase, followed by splitting, which is cytokinesis. And so when you write it out of that, it seems like each of these phases are as important or take the same amount of time as each other phase. But it's not like that at all. If you look at it like this pie chart, you can see that the cell spends the vast majority of its life, all this time here, is in what we would call interphase. And even the name interphase, it's like the phase between when the excitement stuff happens, right? We all learned like mitosis is super important. There's all this drama and excitement, and chromosomes are moving around and things are happening. And in comparison, interphase, the cell just kind of sits there. It doesn't look like it's doing anything. You can't see anything happening with the nucleus. It looks kind of dull. And that's actually kind of the absolute opposite to the truth. So while in terms of dividing into two, not much is happening in interphase, it's all happening during mitosis, which is prophase, metaphase, anaphase, telophase. So if I annotate this, just these parts here are what we would call mitosis. But mitosis is pretty quick, doesn't take very long, and the majority of the life of the cell is spent up here in interphase. And so, why is this so boring? Well, why does interphase look like nothing's happening? Well, during interphase, the cell looks like it's not doing anything, but all the stuff it's doing is on a microscopic, atomic, molecular level. And this is the phase where the cell is actually living its life. It's doing all the things the cell needs to do. So, for example, if this was a liver cell, your liver cells spend most of their life in interphase. And in interphase, they're doing all the liver cell functions. They're detoxifying things. They're breaking down different stuff in your bloodstream. They're storing glycogen. They're releasing glucose. It's really the life of a cell is spent in interphase. It is not boring. It's not like there's nothing going on. It's just you can't really readily see what's going on because it's on such a small molecular level. After interphase, when we go to prophase, then things start happening that you can actually see with a regular microscope. And so that's why people used to focus on that, because you could see dramatic things changing. But really, that's not very significant in the giant overall life of the cell. All right, so interphase itself, now we know a little bit more about what's going on, has been split into these three subphases, G1, S, and G2. These are all the subphases of interphase. And even when we figured out that, we still called this G. It officially stands for gap, like the first gap. And then we do something in S, and then we have another gap, which is called G2, gap 1 and gap 2. So gap, again, is like a misleading term because it implies that there's nothing going on. Really, I think G would be better served by being considered growth. Growth part 1, growth part 2. But that's not the official term. The official term is gap. All right, so let's talk some more about these subphases. So in G1 of interphase, that also is usually probably the biggest amount of time that a cell spends is in G1. And that's when the cell is just being a cell, hanging out, doing what it needs to do. If it's a liver cell, it's doing liver cell job. If it's a brain cell, it's doing brain cell functions. If it's a skin cell, it's doing skin cell things. So, and then something happens in the life of the cell which triggers a transition from G1 into S. And that could be many things. And what this is saying is that the cell is told, you're going to have to divide into 2 soon. So you need to prepare yourself to be ready to divide into 2 cells. That's what happens at the G1S boundary. The cell suddenly triggers something that says, okay, it's time to stop being just a regular liver cell. It's time to get prepared to be divided into 2 liver cells. And we transition from G1 into S. And what S stands for is synthesis. Synthesis means building something. And during the S or synthesis part of interphase, what is being built is DNA. DNA is copied. So in the next part of this unit, we're going to talk about how the DNA is copied specifically. But the fact that it gets copied is still there. And so what happens is usually in your cells, your DNA is looking like this double helix. And you have, if you're a human, 46 pieces of DNA, different lengths that look like this. If the cell is going to divide into 2, we need to copy every single one of those 46 pieces so that when the cell divides into 2, each cell gets a complete set of 46. If you don't copy the DNA, but divide the cell into 2 before you've copied the DNA, then you're going to end up with half the DNA in each of the cells that you make, and that's not going to cut it. You need to have a full set of DNA. So in G1, the cell's going along with its 46 chromosomes, 46 chunks of DNA. It gets the trigger that it's going to have to divide, so it transitions from G1 into S, and in S, all those 46 pieces of DNA get copied. So you temporarily have 92 pieces of DNA, and that's what this little diagram up here is showing. The DNA kind of unzips itself, copies itself, and instead of having 1 piece, which you've got here, you've got 2. They're copied. All right. Then once all the DNA has been copied in S phase, then we flip into growth part 2. And this is when the cell is getting everything else ready in preparation for being divided into 2. So one thing that has to happen is the DNA itself has to be all wound up really, really tightly, because DNA is usually very, very long and stringy and thin, and it can get tangled up, and it's just lots to move around. So to avoid that tangling and avoid that confusion, the DNA is all wound up real tightly. And that's what this diagram here is showing. You can imagine balls of yarn or spools of thread. If it was all just 46 of them, all just unwound, it would just be a big tangled knot. But if each ball was wound up separately into its own little wound up ball, then it's much easier to handle them and move them around without them getting tangled in a big old knot. And the same kind of principle holds for your DNA. If it was all individual strands like this, it would stringy, it would just get all tangled up. It would be a big mess and it would be hard to move around. But if you tied it all up into these big fat balls, you can think of it like that, then it's easier to handle. And that's what happens in G2. Plus all the other stuff to prepare for division. You have to make sure your ribosomes, you've got enough of them, and mitochondria and everything else when you're going to divide it into two. Have you got enough for it to be split into 2 packages and each packet have enough to survive? After that, then we get into mitosis proper, and that starts at prophase. So all these are part of interphase, but there's a lot going on in interphase. The regular life of the cell, then the triggering of the copying of the DNA, and then the coiling up and packaging of the DNA and everything else getting prepared, ready for actual division, which is mitosis. And we're going to deal with that in the next bunch of videos.